Maybe I was going to say, it always takes me a minute to figure out what the view is. I don't know why Zoom makes it really hard to do the regular like presenter view and not share it. Whatever, it is what it is. So greetings, thank you so much for including me in your program. Hopefully I'll give you some useful information about what a cure is and offer some guidance that fits what you're looking for. So my name's Erin Dolan. I'm a professor in the biochemistry department at University of Georgia, national champions. Very exciting. And one thing that makes that especially exciting for me is I'm not just a professor of biochemistry, but I have an endowed professorship funded by our athletic association. So that's pretty unusual that a major university with high profile athletics actually takes the money from athletics and invests it back in academics and research. So some of the work that we do in this space is actually funded by our athletic association. Yay. Okay, enough of a plug for University of Georgia. It's just been a special week for us. And instead, let's think about undergraduate research experiences. Obviously, you all are bought into this or you wouldn't be part of this session. But I just want to sort of call to mind a couple of resources that might be of interest if you haven't heard of them already. So we're all in the life science space. How many folks are familiar with vision and change? Like raise a hand or type in the chat. Okay, so I'm seeing some folks who are not putting up a hand. Great. So I'm going to bring this to your attention. Vision and Change is the sort of guiding document for what undergraduate biology education should look like. And it came out in 2011. I guess I've got the date right there. And so lots and lots of folks across the country who are involved in teaching undergraduate biology students use this as a framework for thinking about what students should learn. And one of the interesting things in that framework is that it advocates for research experiences as an integral component of biology education for all students. So I assume you all, I know it's awesome to me, it's awesome. I assume you all teach or work with undergraduate biology students? No, okay. Tell me a little bit about what this group is, just so I'm on the same page.
Most of them do,
okay.
But there are also high school teachers here.
Oh, yay, cool.
And there are students.
Oh, yay, that's even cooler. Right on, thank you for that. Okay, so for people who are at the high school level, you're maybe preparing students for this, and for students, you may actually be involved in it. For folks who are at undergraduate institutions with biology students, how many students do you have? Maybe type in the chat. Or in your high school research class, like how many students do you have? Okay, so I'm seeing good numbers. And think about not just maybe how many biology students are there at your institution, or how many biology students are taking, or students are taking biology at your institution. Oh yeah, now we're starting to see the big numbers roll in. So maybe an individual class, but now we're starting to see some of the big numbers roll in. So this becomes a real challenge, right? We're thinking about, all right, if all biology students at the undergraduate level should be involved in research, this becomes a really big challenge. So why is it that people are advocating for this? And you can either unmute or you can type in the chat. Why do we want students to participate in research? How do they benefit? Either think of students you work with on research or think of your own experience as an undergraduate.
Application of what you've learned in a new environment.
Great,
like
so you get to apply what you've learned.
Yeah, everything up until that point is kind of training wheels on.
Yeah, information goes in, but then you actually get to do something with it. So real world problem solving, I'm seeing ideas in the chat. Feeling like you're part of the scientific community, identifying as a scientist, learning that failure is normal and troubleshooting, how to troubleshoot, learning teamwork, problem solving. It's the actual experience of being a scientist. Y'all are hitting on exactly the reasons that have motivated some of these recommendations to involve many, many more students in research. They fit into 4 big categories. So first is knowledge and skills. So students obviously learn stuff and they learn how to do stuff by participating in research. Students also develop certain attitudes and dispositions. So for example, they may see, oh, like that's what research is about. Or you know what, even if I don't want to grow up and be a researcher, I at least see the value of it. So I see research as a valuable endeavor that maybe we should spend taxpayer dollars on. It's a way for students to develop identity and connections, and this came up in the chat, starting to feel more like a member of the scientific community and connect with people that they would not otherwise connect with, or in different ways than they might otherwise connect, such that they expand their academic or professional network. And then finally, research experiences are important for clarifying education and career pursuits. So deciding, do I want to do research professionally? And if so, how do I go about that? So then the challenge becomes who gets access to research experiences. And I'm going to focus at the undergraduate level because we're talking about cures, and the U stands for undergraduate. Although I do encourage folks who are at the high school level to chime in. Think about how students get access to research experiences. And you can either think about how students get access to doing research with you, or if you did research as an undergrad, how many people did research as an undergrad? So how did you get access to that research experience?
Oh, you want us to answer?
Yeah.
Oh, I went to a professor and asked if I could work in his lab.
Great. So you went to a professor and said, hey, say that meant you knew what the research was a thing, and then you asked for it. Mm-hmm. Jan, you want to chime in?
Hi, I just typed it into the chat.
Great.
I teach a molecular biology research course that is purposely designed to teach students how to do research. So they just spend the first 2 or 3 weeks learning how to do DNA barcoding, and then they all do their own DNA barcoding project, which could be a project that they design, or I have some collaborations with researchers at other institutions, and students of mine in my class can do a project that contributes to those larger research efforts.
Nice. So you have a course where it's embedded. Several folks put in the chat that you just kept hanging out and somebody eventually asked you to do some research. So you were sort of in the right place at the right time. So some degree programs required it. Kate's noted that her degree program actually required it. Our degree program in biochemistry requires 2 semesters of research, but that is not common given the number of students. And then several of you point out the idea of an internship program. So you apply, those are competitive. And what ends up happening is that for especially research internships, I'm not talking about courses here, I'm not talking about cures, but especially research internships, which is historically how students participate in research. We get access to research via, in a way that can be explained by Bourdieu's theory of capital. And I want to talk folks through that, so you're sort of thinking about that as you think about engaging students in research through your cure. So we know that people get access to limited resources, light research experiences, using 3 main forms of capital. First is human capital, and that's the idea of the what you know. So that might be the coursework you've taken, the grades you've earned, the test scores students get, prior experience even. So let me tell you, the students who have access to a research experience in high school, amazing, not common. But it really, those students then stand out when they get to college. Then there's the how you know. And I saw in the chat somebody posted, I just asked a lot of questions in class. And that's sort of this how you know. If you ask a lot of questions, then people who are in science are like, you ask a lot of questions. That's what scientists do. Come be part of my research. I'm a good example of this. And I see that not everyone's from Pacific Northwest, but a number of you, it looks like, are from the Pacific Northwest, because I'm seeing typing related to that. So I actually grew up in Seattle. My dad, after working in industry for 20 years, became a faculty member at the University of Washington in engineering. And so my parents were always overeducated, and my dad's friends were all faculty. And so I just kind of hung out with a bunch of STEM professionals, and it was just normal. So it didn't scare me to go up to a faculty member and ask for a research experience. I knew it was a thing. So I had that kind of cultural capital. And there's finally the who you know. So were you hanging near a faculty member who then gave you an opportunity to participate in research? Are you part of a major and that's required for participating in research? What we know is that students who come to college with these three forms of capital get access to research experiences. And of course, because they get access to research experiences, they develop additional capital. They learn more stuff, that human capital. They learn more about how scientists work, so they seem in the know, that cultural capital. And they also develop social capital. They make connections that they would not have otherwise made had they not participated in that research. That helps them get access to additional experiences. So what that means, especially at the college level, is students who come to college primed for careers in science and in research are most likely to persist and succeed. That means that we really need to think about doing something different to change the status quo. So it's not just like me, kids of faculty members who then grow up to be the next set of faculty members or the next set of researchers. And so this is really what cures are about, is how do we broaden access to research experiences and achieve these outcomes for students. And I'll give you the definition that I like to use for cures, although I'm sure folks would argue with me. I firmly believe that a course-based undergraduate research experience is when a whole class of students, so everyone involved in the class, addresses research questions or problems that are of interest to the community outside of the course. So that means that there's a research question or problem, or more than one, that students are addressing, and that the results of their work are of interest to someone outside the course. So that can be, I think I saw in the chat some idea of like local water resource groups or civic groups, biotech, academic researchers. It doesn't matter who that group is. The point is that it matters outside of the class. It isn't, the purpose isn't solely limited to the class. So to help us think about this definition, I'm going to give you a couple of examples of projects that students might do in a lab course, and I want us to think about whether each of them is a cure and why or why not. And actually, I'm going to skip over this. Okay. So the first example, okay, so I see a question from Uwe. What role can it play, to what extent a research question is of interest to the students? That is a great question. We don't know. No one has studied that empirically. We're working on that right now. I think that there's lots of ways to give students choice and ownership, and that can happen even if the students don't generate the research question themselves. So we know that. The students don't have to generate the research question to develop a sense of ownership, but we don't know the influence of generating the research question. I personally, in my cures, don't let students generate the original research question because they don't know enough to ask a really good investigable research question. That doesn't mean they can't formulate plans or make decisions related to troubleshooting, to problem solving, to how to analyze the data, to what to do next in the research. But I set the boundaries for the research because I know more than they do, and I know what's a good research question. Jan, go ahead.
Oh, hi. I just wanted to say I kind of try to combine both in my class, both what interests the students, but also focus them. So hence the focus on DNA barcoding. And we talk a lot and do some reading about how DNA barcoding can be used in so many different ways in research in conservation biology, in the medical world, looking at viral infections or what, so many ways. But then students either design their own project about something that's interesting to them, or take part in these collaborative research projects that I have going. But it's all focused on DNA barcoding for the same reason that you've said. You can't really handle even a small group of students that are all doing something completely different. So they're all focused on a set of techniques and one sort of question that can be applied in a bunch of different ways.
Great, thank you for sharing that. Okay, so I'm gonna give you 5 scenarios and I want you to chime in either aloud or in the chat. What if students are addressing a research question that is unknown to them, but the research community knows the answer? Would you all think of that as a cure or no?
I mean, that's like
Happens in real research all the time.
I'm interested, I'm seeing a lot of no's, so I'm interested to hear what you think, why you think that. So someone who said no, why not? Why did you say that?
Because they could research this and find the answers themselves, which should have been the first step, which is usually the first step in a cure.
Exactly. That's what you hope, right? So that people look at what's already known. Sometimes people have a lot of confidence and don't think that. I always assume in any research that I'm doing that someone else has thought of an answer. And it's only once I've done all that reading to make sure no one else has addressed that question that I embark on a research question. Right. So I would say no, because we already know. How about this? If students only come up with thought experiments that are novel, so they don't actually do any experiments, they just propose them. Okay, again, I'm seeing a lot of no's. Why not?
I think somebody that I can't remember who said this, but they were basically like, ideas are a dime a dozen. The important part is to actually do them. Anybody can think of any idea, but you've got to be the one that actually figures out how to follow through.
Yeah, and the figuring out is actually part of the process. I do see the can be though. So Todd, would you be willing to elaborate a little bit on the can be?
Yeah, I think sometimes a thought experiment is the basis. So it's kind of how you follow up with with that, and don't just dismiss the idea. But is it reasoned? Is there evidence? Is there, all of the pieces that you need to do that next step?
Yeah, so there's definitely part of the process here, and some fields are based entirely on thought experiments, like theoretical physics. But for the most part, a thought experiment is not sufficient, right? We really need students to follow through so that they get the actual boots on the ground experience. Okay, here's another example. What if students identify what bacteria are on their shoes? Vote in the chat, yes or no. Hey, David, I didn't see you were on this. Down the hall. So I'm seeing some no's. How about why no? And then some yeses and why yes. So someone who said no. Okay, Kate.
David. So there are programs where people go out and find bacteria, like the Tiny Earth Project. So that has a purpose. They're looking for new antibiotics. And
I love that you mentioned that, David, because that's exactly it, right? It depends on what the purpose is. So, and you see that Uwe mentioned Chernobyl. It depends on whether there's a purpose to doing it or if no one else cares, like Kate mentions. If they're just doing it to see what bacteria are on their shoes, that becomes a little bit of like a middle school science fair project. It's not really addressing a question that matters outside the classroom. And instead, what we want to do is actually provide that purpose. So I'm glad y'all brought up that word. Okay, what about this? If students analyze a question that's unknown to the research community using a pre-existing data set. Okay, I'm seeing a lot of yeses. Why? Talk about why yes.
There's new things in existing data, and it requires novel approaches sometimes. Sometimes it requires like obvious things that nobody's done.
Absolutely. In fact, some whole fields are based on this, right? Using pre-existing data sets like bioinformatics. You're not actually going out necessarily and collecting those data. You're using data that already exists from other sources. So this is sometimes hard for students and also hard for instructors because they think, oh, the students have to collect the data. Well, we don't actually know what uniquely students benefit from collecting the data versus using pre-existing data. Data parasites of words, I love that, thank you. Okay, last one. What if students characterize novel versions of a protein of interest to the scientific community, but get negative results? Yes or no? Seeing a lot of yeses. Why yes? Somebody willing to say why yes?
They're still doing all the experimental work and analyzing what they got and building those skills of trying to figure out, well, maybe why did it not work and what can we do next to, redesign and try again.
Absolutely. This is absolutely part of the research process. Now, this is something that's challenging for students. Sometimes they will conflate getting a negative result with failing a grade. Like a failed experiment becomes a failing grade. And so you have to manage their thinking about what counts as failure in research versus what counts as failure in academics, and really focus on that process instead of the product. So I really want to call attention to two aspects of this that hopefully became clear that makes a cure distinctive. So first of all, it's focused on the process of doing research, and that there's always a purpose, that the work that's happening in the classroom matters to others outside of that classroom and is communicated to the others outside of that classroom. And this strict definition is super important if we're arguing that we're increasing student access to research. They have to be conducting research if we want to make an argument that this can count as a research experience. Okay, so how to do this? I'm going to give you just a couple of pointers before we wrap up. So very first thing I find is critical is to start with research goals. And Sandra asked me to comment briefly on an example of a cure. So many moons ago, folks, are you familiar with Arabidopsis? How many people know Arabidopsis, little plant here? Okay, like this is the fruit fly of the plant world. So it looks like a lot of familiarity. For folks who aren't familiar, it's like the model plant that we study instead of studying plants that are larger and more difficult. And back in, 2000, its genome was sequenced. So we knew all the sequence of all the genes. And in 2000, yeah, there you go. Exactly, a big source of existing data. So the working hypothesis at the time was, let's knock out every single one of the plant's genes and see what happens. And groups of scientists funded by the National Science Foundation, it was actually called the 2010 project, because we were going to do this all by 2010 and figure out how plants worked by the year 2010. Oops. Knock out their family of genes that they were interested in one by one, and then grow the plants and see what happens. So they could figure out the purpose or the function of every single gene. Okay, so knocked out a lot of the genes, and only 3% of the plants showed a phenotype. In fact, there's a paper called "Dude, Where's My Phenotype?" that you can Google and find. So why is that? What could be going on?
Or one way to skin a cat?
Yeah, so maybe it's redundancy. And they started knocking out families of genes. And in some of those cases, didn't see phenotypes. But you're right, that's one working hypothesis. So what else could be going on?
They didn't germinate?
So definitely there's some lethals. And that's, again, a fraction, but not a huge fraction. So what else could be going on? What if the gene's important for the plant's ability to respond to herbivory? I'm giving you a little hint there with those spider mites. What would you have to do?
Unless you actually introduce whatever environmental stressor or response mechanism, you won't actually see the phenotype. Like, for instance, if you did a knockout of a mouse for anxiety, but then you didn't put them under any stress, you would never see a phenotype.
Exactly. So how many different stressors do you think plants respond to in their environment?
One.
A jillion? Thank you. That's actually a number, a gazillion, jillion. And so this becomes a math problem. So Arabidopsis has 25,000 genes, more than humans. And then a metric ton of environmental stressors to respond to. But this is the cool thing about cures. A whole class of students can take their mutants or focus on a set of genes and actually generate those environmental stressors, grow the plants, and report their findings back to the scientists who are interested in those genes. And in fact, we set up a project called the Partnership for Research and Education in Plants for undergraduates, where partner scientists work with classrooms specifically focused on plant herbivory. And the reason that we focused on that is that the plant scientists we were working with didn't want herbivores in their labs, but they were fine with them being down the hallway in a classroom. So the groups of students could tackle characterizing gene function, and some of their results were actually included in papers or as preliminary results in grant proposals. And it allowed the plant scientists to gain some insight, unique insight into their genes of interest without actually putting herbivores near their stock plants. So that was the research goal, is to characterize the function of genes and the plant's ability to respond to herbivores, and also the role of, or basically how feeding on those mutant versus wild type plants affected the herbivores' responses. Once you have research goals, then you can think about student goals. And I'm gonna skip to that. Because of course it's a course. I encourage folks to think about student goals that matter beyond the end of the course. So you'll see we have some examples here, including designing a well-controlled study that builds on prior knowledge and makes progress toward a research goal, making and defending decisions during experimentation, and communicating about research progress orally and in writing. So this sets the learning frame and then the research frame. And then we used objectives, sorry, I'll skip this, to connect the two. How many folks are familiar with backward design? Have folks heard of backward design? A couple of folks. So this is the idea where you start with the goal in mind. You think, where do you want your students to go in terms of both the research and their learning? Where are they now? And how do you connect them? How are you going to get the students there? And the way to think about research objectives is to think about measurable, observable things students will do. For example, to design a well-controlled experiment that builds on prior knowledge and makes progress toward a research goal, you need to know some prior knowledge. Happy to share the slides. They need to know something about the Arabidopsis life cycle. They need to think about, what do we know about the genes that are already involved, and so on and so on and so on. And the cool thing about this is that once you've done this laying out of objectives, you actually have a plan of action for your cure. So that's the way that I recommend going about teaching cures, and that's one example that I'm happy to share. There are other examples in other places, including large cure projects like the Freshman Research Initiative at the University of Texas, vertically integrated projects at Georgia Tech. There's examples of both introductory and senior level cures. There are mini cures. One was mentioned earlier, the idea of prevalence of antibiotic resistance in the environment. UC Davis has a series of first-year seminars. And then finally, there are large cross-curricular cures that are examples. So all of those are examples you can look to, as well as the CureNet website. And with that, I will close. Thanks so much for your comments and your questions.
Thanks, Erin. I think we have time for a couple questions, and then we will have to say thanks. And I'm going to stop the recording now.